a quick zoom.
Okay. Come on in, video. We'll do it from here. Thank you.
I don't know if Chris Maz is joining us from the Smithsonian Institution. He's our expert on these sea stars or asteroids. Not very abundant at these steps, but they are considered what's called. No, I think that was good enough for an ID.
Okay. I saw a peculiar white little cup-looking shape over here.
So yeah, they're considered, even though they're not abundant sea stars, they're considered often keystone species, being very important to the ecosystem and to the communities. So having a disproportionate large effect on the communities. That's disproportionately important to their low abundances. And so there we see our first scleractinian coral. That's a scleractinian cup coral.
Is it dead?
Yeah, so it looks like we only
have the skeleton. Well, maybe not.
Hello, Chris Mock from the Smithsonian here.
Excellent. Welcome, Chris.
Hi, Dan. So yeah, that white sort of nondescript sea star, it's definitely a Goni asteroid. It looks like either Mediaster or might possibly be something like Gilbertaster, something a little sort of off the beaten cuff, unusual. We don't know a lot about them. They're usually only taken from trawls anecdotally. I mean, one or two of them. They're probably just deposit feeders, probably making the best they can of whatever organic material is living on the sediment in that area. But we don't know a lot about them. And so even living observations is really useful. And knowing that they're, for me anyway, knowing that they're, white to yellow is like one more fact that I knew about some of those yesterday. So I appreciate that you got it. Any one of these,
I'd like to request a ship
sometimes literally 100% more information than what we knew beforehand. So.
Excellent. Thank you so much,
I'll
Chris.
continue to monitor. Thanks.
Good copy, Bridge.
Yeah, so that was Dr. Christopher Ma joining us from the National Museum of Natural History, the Smithsonian Institution in Washington, D.C., our expert on sea stars as well as other echinoderms.
I don't hear anybody chiming in. Okay. Okay.
And so this...
I'll just continue on. I don't know if... I don't think... Didn't sound like there was interest to get a closer look.
Yeah, maybe a quick zoom. I'm sure Chris will appreciate it.
Okay, sorry. My mistake. Okay. Stand by 1 more second for me, sorry. Just trying to
So,
get it tricky to
looks
hold.
like
Come on in.
possibly in the genus
Take
Dictyalis.
the camera, co-pilot, move it wherever they ask.
Copy that.
Move up a little, get a good view of the
Yeah,
web.
so I thought.
Hi, Daniel.
Sorry, Chris, you broke up there. Can you repeat?
My best guess would be Dictyalis also.
Excellent. But different to the ones that we imaged on last year's cruise. Yeah, I just have the photo up here. And so those are also hexactinellate sponges or glass sponges. So most folks, when they start seeing these images of these deep sea sponges, they might be bewildered by the different shapes and all these kinds of very unique looking features of sponges. A lot of that is that these are fundamentally different. They're in a completely different group of sponges, the hexactinellid or glass sponges. Those you typically do only find in a deep sea. There's only a few species that are found in shallow waters. And they're differentiated by the shape of their skeletal spicules. They're siliceous skeletal elements, and they have this either four-pointed or six-pointed shape that are fused together. And so in the glass sponges, you kind of see it's a very glassy and rough surface, and they're particularly porous.
Tilt down a little.
And looks like we might
just want to get the whole thing squared up in the middle of the shot. There we go.
Turn lasers off.
Sure.
So beautiful view here of this sponge. Best guess is that's in the genus Dictyalis, which is in the family Euplectellidae.
Thank you.
Thank you.
Lasers back on. Thanks.
So we're at a depth of 1,264 meters. We landed just off this small ridge crest that's on the southern side of this seamount. That up until yesterday we had no maps of the area, and yesterday we were able to map the entire feature. That was one of the benefits of having extra time on these last couple of days, is that we've been able to cover huge gaps, but luckily the weather has cooperated for a dive today, and luckily the crew has agreed to give us a long dive today, and so we'll be staying here on the bottom till 6 o'clock Hawaii time.
So as we travel up this ridge, we're actually traveling perpendicular to the main rift zone ridge axis where the volcanic eruptions typically are sourced from. And so what we are kind of looking at is we're looking uphill towards where many of these eruptions would have been coming down. So just like water, lava likes to flow downhill, and we're kind of looking up the hill that it should have been flowing down. So as we go up this ridge top, we might be able to make out individual lava flows as these parallel layers that are kind of stacked up like the layers of a cake, but they're going to not be horizontal. They're going to be dipping into the direction that we're looking, and gives you an idea of where the lava flows were actually flowing at the time that this seamount was an active volcano. And so many of you may not have known this, but almost all, if not every single feature that has elevation within the ocean floor was generated due to volcanism. And so volcanoes are a primary source for constructing the oceanic crust and the features that are actually on top of the oceanic crust. There's also been some really interesting landscape structures that seem to be like channels, and which have concentrated a lot of loose rocks and sediment throughout the entire time. That's because currents are sweeping through the surface and letting loose rocks and sediment deposit within the channel.
And so, yeah, one interesting observation throughout the dive. So we just landed on the bottom about an hour ago, and so we landed just east of this ridge, and there was a few fish there, but few corals, and then we moved towards the ridge, and are moving along the ridge crest, and there's nice moderate density of animals along this ridge crest. The dominant group of corals here are the primnoid corals. So yeah, these bushes that you see around us all appear to be the same species.
Get in front of me here.
And
Copy that. Co-pilot.
few sponges. We've seen a couple of these stalked sponges. We're just past one to our left. Those are in the genus Okeanos. So yeah, throughout these few dives we've done on this expedition, there seem to be kind of depth zones or kind of and where different groups of corals are the dominant ones. So here we are, this primnoid coral dominated assemblage, and we saw a similar one on one of our dives. On some of the deeper dives we see the bamboo corals being more dominant. So it seems to be some kind of stratification in terms of what families are more common in different areas.
So as you can see, the surface of this seamount, being extremely old, has succumbed to gravity and erosion over the long lifetime that it's had on the ocean floor, and you can make out a lot of rock surfaces that are starting to crack and fall apart. So even if it's heavily cemented by manganese crusts, time will eventually take its toll on these volcanoes, and they tend to break apart and succumb to forces of gravity as they form these nice talus slopes that we've been collecting rocks on over the past couple dives.
Left corner is your mini Zeus that's looking up the hill. Look for
if we have a quick second in the area, can we just get a quick zoom on one of these corals, and in particular on the associates of it?
Yep, absolutely. Straight ahead, Wesley?
Yep. Either one of those two. Actually, the one just off to the right of the lasers.
To the right?
Seems to be a bit more.
Right, video, if you want to come in.
Roger that.
So yeah, these are all primnoid corals.
If I start to drift, you can take my camera.
And you often see these ophiuroids perched up on top of the coral. And a lot of times it's a symbiosis that's mutualistic, meaning that both the brittle star or the ophiuroid and the coral benefit from this living together. The brittle star gets a habitat somewhere to live, but it also will eat and clean the coral, cleaning its tissues from parasites and other things that might grow on it. So kind of a win-win living together here. Good roommates.
Guys, we have a visitor in the ECC right now. Michael Garcia is with us.
Oh, welcome, Michael. Thank you so much for joining us.
Thank you very much. It's wonderful to be here and to see what's going on. Thank you.
Excellent. Yeah, and so you're probably going to be excited that we did collect a rock sample there, a nice angular rock there at the beginning of the dive. So yeah, on this seamount that had never been dredged, never been mapped actually, and so yeah, it's going to be of interest, I'm sure, to you and your colleagues.
Sounds terrific. Thank you.
And yeah, on that close-up of this primnoid coral, we got a nice view, and Andrea pointed it out, that we have these downward-facing polyps, and that's characteristic of this genus, the Norella. And Christopher Kelly from the University of Hawaii, also science elite for this 3-year effort, provided the ID. This is Norella dichotoma. Dichotoma in relation, or referring to the form these corals branch, so they have dichotomous branching. So in every one of the branching points you see one branch going into two, and so that gives it a very characteristic kind of shape. And so the branching pattern is one of the features that is typically used with a lot of corals to differentiate them. Now, one problem with having such a character is that branching can be somewhat variable, and then in particular with young colonies that have not yet branched, it doesn't allow you for a way to identify them, and that's a common thing is when we see very small colonies, it becomes difficult to differentiate. So all coral colonies start with a single polyp, and at that point it becomes very difficult to differentiate them. But coral taxonomy in general is a very difficult thing. There's a lot of plasticity, so meaning that you could have the same species starting to look very different if you have it in different environments, and conversely you could have different species starting to look very, very similar if they're in the same environment. And so what characters you use to differentiate the species are difficult. Increasingly scientists are using DNA molecular technologies to differentiate corals, but that comes with other suite of problems. So it's still a work in progress, and there's a lot of active research in this area. Is how do you differentiate the different species? Depth is 1,235 meters. Maybe we can take a quick look on that pinkish looking coral towards the right. I don't know if that's different. A small colony up on, yeah, up and right. So that looks like a pink coral, possible genus Hemicorallium. I'll take a look here. And Chris has just ID'd that sponge too on the lower right, and the genus Atlanticella.
So where is it, Washley?
Just about a meter above the lasers and a little to the right. Just the only pink object there in the view.
Yeah. Just try to sit down.
And we see a few fragments here of dead sponge material.
You can look at your nav cam, see your
so you can see how these kind of like brownish structures which are amongst the rocks there. So after sponges die, they will in a lot of these places you see a lot of dead sponge material. And it's interesting that still at that point there's different organisms living on top of the dead sponge material. So that's the real importance of these habitat-forming species like corals and sponges, is that even after they die, they provide important structure to the environment and habitat for a myriad of associated organisms.
And as we stay zoomed out here, you can kind of make out a slight tabular shape into the rock that this pink coral and is sitting on. This is kind of what I was talking about earlier, where you can make out the actual direction of the lava flow and the way that it's dipping. So it's dipping towards us. We're looking up the ridge at the primary rift zone ridge where this lava most likely would have been sourced from. So the eruption would have taken place further up the ridge, and this tabular shape to it kind of tells geologists and myself that this is one of the two types of lava flows that we were looking at or talking about earlier, which is called a sheet flow. It has a tabular structure rather than a globular structure.
So yeah, this pink coral that we were looking at, that's Corallium is a family, and that's going to be in the genus Hemicorallium. Possible Hemicorallium abyssale. Pink corals or also red corals are important. They've been used by many cultures to manufacture jewelry. In fact, humans have a long history of using pink corals for jewelry. There's remains of pink corals being found with the Egyptian in Egyptian cultures and ancient civilizations. You might ask yourself, how did they get access to this? There are a couple of species in the Mediterranean Sea of these pink corals that are found much shallower within scuba diving depths. And so for a while there was also a fishery for pink corals here in Hawaii. That fishery is now dormant. But yeah, in many cultures, pink corals have been used, and it's the skeleton. So after these corals die, the skeleton has this bright pink, shiny, lustrous appearance, and it's
set yourself off to the right there.
Yeah, I'll port over.
My heading is getting pushed left by the current quite a bit, but so you don't have to come all the way over into my view. I've been reaching my set point right to try to compensate, but I'm still getting blown off. So
Copy that.
Try to make our way here up the ridge, cover some ground maybe. So we're current depth is 1,235 meters. We do hope to climb to the very top here of this ridge, which should flatten out around 700 meters, so about 600 meters of elevation gain. And in the process, we'll be going straight through the oxygen minimum zone, and it's going to be interesting to see how that shapes the biological communities. So far in the dive, we've seen a nice moderate density in some places, primarily dominated by these primnoid corals, and that in the genus Narella, at least a couple species, but one Narella dichotoma. And I also want to take a second here to once again welcome and thank all of our shore-based scientists for joining us, and not just today, but also for being active participants, even when we had weather days writing in. So thank you very much. We have a nice group of engaged scientists, which are really making this expedition get a lot more out of it than what we could do single-handedly. So yeah, thank you so much.
So Dan, quite a crowd that's developed here at UHCCC. We have Les Watling here, Patty Fryer, who's going to be on the CNMI cruise, and Jeff Drazen here as well. So we got a nice group here participating.
Excellent, yeah. So welcome and thank you for joining us.
Bridge, this is Nav.
So yeah, University of Hawaii, we have a nice group of experts, a lot of
I'd like to request a ship move, same as last time.
Experts who work on this deep sea realm, working here in Hawaii.
0 degrees at a speed of 0.2 knots.
And so yeah,
So that move's going to come at you.
So we have here on the ship, as Jonathan and myself are the ship-based scientists, but then throughout the country there's several what called exploration command centers. So there's a series of monitors, and where scientists are watching the video in real time, the same time we should see it here on the ship, and they're connected to us via teleconference line, and really allowing us to get a lot more out of these expeditions that you could do just with a small group. We have that way we can have a much broader scope and expertise, people focusing and being specialized on different groups, all participating in the expedition. And then another, of course, aspect of this telepresence technology is that the general public can also enjoy these images and watch it in real time and join on the excitement. So this is exciting for many reasons. I mentioned that this seamount is close to 13,000 feet in elevation. So if it were above the surface of water, that would make this third largest mountain in Hawaii. Just a little shorter than Mauna Loa and Mauna Kea, but up until yesterday we had no maps of this seamount. And so yeah, it's just unbelievable to think that a lot of these even enormous topographical features are still unknown, and we don't even have maps for many of them.
And not only do we not have any maps, we also don't have any geological samples, so that was another monumental achievement that we've already gotten a geological sample from this seamount. This is going to reveal not only how old the seamount is, but a whole lot of other things about the history in which, the geological history in which this seamount came to be, such as some of the stories that the crystals and the chemistry of those crystals can tell us about how the magma chamber evolved, or what kind of pressures and temperatures in which the magma that supported this construction of multiple lava flows. And as Dan said, so 13,000 feet of piled up volcanics is what is making up this seamount. And so now that we have a geological sample, we can definitely start to do some more in-depth investigations in the laboratories once we get that back to the shore-based scientists.
And then give it a little more thrust to pin yourself. You don't need to come in and hammer in. If that makes sense.
Yeah.
Yeah, so I mentioned that seamounts continue to be discovered. We know of about 10,000 around the world. Very abundant, and because of their abundance, they're considered one of the most important oceanic habitat features. You typically have higher abundances of life on the flanks and summits of these seamounts that you would find on as compared to the abyssal plains off to them.
Okay, video if you want to come in for partial.
So you're up.
Thank you, co-pilot.
So here we have another one of these primnoid octocorals. Also looks like in the genus Narella, these characteristic downward-facing polyps. And an ophiuroid, again, this very common symbiosis between the brittle star and the coral. So I was talking about seamounts and the importance as important habitat, and I said that we have about 10,000 of them, but we still continue to find new ones. Some of these seamounts you can detect with satellite altimetry. So with satellites, you can take accurate measurements of the sea surface height, because there's kind of small, on top of these seamounts, you would have piling up of water, and so you would, with satellites, be able to measure that the sea surface height is slightly higher on top of these seamounts than on the surrounding ocean. And so if you take a measurement over the entire world, you could see places where they might be seamounts, but then in smaller seamounts you don't see that as much. And so yeah, on this expedition itself, we mapped 4 new ones that had not been mapped, including one that didn't even show up that well in the satellite altimetry. So it's yeah, very interesting that we continue to find these enormous features.
Got it.
So I just want to add a little bit to what Dan's saying and provide an explanation for why the water, as he said, piles up over seamounts. So everybody is more or less familiar with the force of gravity that keeps you attached to the surface of the Earth. Well, the gravitational force actually changes ever so slightly whether there is a excess of mass under where you are currently located, or a deficit of mass under where you're located. And so these seamounts, you can think of them as a slightly bit more of mass, having a slightly bit more gravitational attraction due to the excess mass. That gravitational attraction is what brings in more water from the surrounding areas, giving it a slightly different elevation of water. And this is what satellite altimetry is measuring. It's measuring the relative differences between the satellite and the ocean surface. And over these seamounts where there is an excess mass because there's a big mountain under the water, it's pulling the water to it more strongly.
Looks like the ship finished its move. A couple more minutes of swing on you boys.
Thanks.
We're going to take a quick zoom on this animal right here, which is the first time we've seen this on this dive. So far, this community seems to be dominated by a nice moderate density of primnoid corals.
A minute or two here, so.
Yeah.
And again, you can kind of make out some faint layering going on there that is dipping from the left to the right. These would be the lava flow layers that are flowing off of the rift zone ridge above us. But as you can see, as we zoom in, you can kind of see the botuloidal texture that I was talking about, or that globular texture of the manganese crust, that precipitate that forms on top of the rocks. That precipitate is starting to grow and grow and grow as the rocks become older and older and older, concealing any of the primary textures left over from the volcanic eruption themselves. So it's kind of hard to see a lot of the primary structures, but we have been able to make out some layering within the lava flow structures themselves. So that's been a neat thing to see in the changing in landscapes that we've observed so far in the dive.
Yeah, and so that looked like a mushroom coral that was just closed up. And I believe because of the long stalk, it might be a small one of these Anthomastus tahinotus. So you could definitely see the longer stalk and the closed polyps, the retracted polyps. So yeah, some corals are able to completely retract their tentacles into a groove. So octocorals have this ability to do that. To our right is one of them also conspicuous sponges. That's a Phorionea or Oca erecta, and on top of that rock there looks like maybe our first bubblegum coral of this dive. And we can do a quick snap zoom on that kind of pinkish coral in the distance there.
This one, watch it?
Yep, exactly.
All right, video. Gonna do a quick zoom.
Sure.
Yeah, and so this is a bubblegum coral, or Paragorgia.
Nice work.
And then bubblegum, a common name for this species is, because as I mentioned, so corals, or some corals can retract their polyps into a groove, and when the tentacles are completely retracted, the rest of the branching kind of looks like chewed up bubblegum. There's several species of these around the world, including some that are very massive off New Zealand. There's one species of these that can reach heights of 5 meters or more. And yeah, very important, including some that are very massive off New Zealand. There's one species of these that can reach heights of 5 meters or more. And yeah, very important again because they create so much structure. They create a lot of habitat for a lot of fauna.
Yeah, nice.
All right, onward.
Depth right here is 1,220 meters. So yeah, we absolutely want to make it to the top of this ridge. We're diving on this south ridge. The seamount gets to within 650 meters of the surface. It's kind of a flat plateau at the top, around 700 meters. So we absolutely want to make it to that top throughout today's dive. We do have an extended bottom time, so just to give all of our viewers a heads up, we are not going to be leaving the bottom until 6 p.m. Hawaii time, so we should have a nice extended dive today, hopefully to make up for at least a little bit of the time lost during the last few weather days.
Okay, you're getting pushed out.
So the manganese crust that we've been observing so far on the dive. It's pretty thick in local areas. However, I think that the extensive erosion on the surface of this seamount has exposed some really neat landscapes along the dive so far. So we've been able to make out some layering under the manganese crust that suggests that these are different lava flow layers.
Like a look at. We have a little time here.
There's been some concentrations of sediment and talus within what appeared to be some small channels that have developed on the surface of the manganese crusts.
All right.
Pilots, you ready for a move?
Yeah.
Again, 30 meters, same as last time.
So yeah, we just
045, I would
called
say.
in another ship move, so we have a couple minutes here in this area before we have to move.
This is nav.
It's a very interesting way that the moving happens. So it's actually a three body system. So you have the ship tethered to a Sirius, the first ROV that hovers just above the substrate around 50 meters or so, and then you have a second vehicle that's on the bottom, the one that you're seeing the images right now. So when the ship moves, it kind of moves like as a caterpillar, that three-person team. One moves, then the other vehicle moves, and then the third vehicle moves. So it takes a lot of coordination. And then, of course, you have a lot of cable. There's about a mile of cable between the ship and these vehicles.
And so Dan was mentioning that we have two bodies of the ROV. There's the Deep Discoverer and Sirius. Sirius hovering above Deep Discoverer D2. The live video feed on camera 2 that you guys might be watching in the lower left-hand quarter panel shows the view of Sirius looking down on D2, and usually is capturing some beautiful scenery from the entire landscape. You can get an appreciation for the three-dimensionality of the terrain that we're diving on.
So yeah, this is another primnoid coral. In the background we see the long-legged shrimp, and that's also very conspicuous. It's going to be in the genus Nematochircinus, Spanish tenor rustris. And you see these very commonly down at these deep depths.
Just a bit wider shot.
Hi, Daniel. This is Andrea. I was just going to make a comment about this primnoid. That close-up, you could really see those small scale-like structures known as sclerites. They're made out of calcium carbonate, and you can really see them. They're fairly large in primnoids. They overlap. So it's a really great image.
Excellent. Thank you. And yeah, Andrea, so the video is absolutely spectacular. You see a little gastropod there to the left. So yeah, you see a lot of these animals that are very small, and then you see small structures. So a lot of time these fine things that typically you need a microscope to really appreciate and see, you can see with these high definition cameras, and it's just absolutely phenomenal technology. And then yeah, also our talented crew. It takes a lot of coordination between the pilots and the videographers getting these beautiful images, even in higher current environments. And then to think that these vehicles are about, in some cases, about 1,000 miles away from where the scientists are viewing this in Honolulu, about a mile deep, and you can appreciate this very high quality product. It's unbelievable technology. So yeah, a lot of primnoid corals. Also in between we see a few of these pink corals and bubblegum corals. And there's an interesting looking sponge.
Can we look at that blob?
Yep. Yeah, we'll take a quick look at this sponge.
Copy that.
Oh, and I think there's a fish to our right. Maybe before.
What would you like first? Fish or sponge?
Maybe the fish, because it's probably going to move away. So that looks like another rat tail or macrurid. Grenadier is another common name for that group of fishes.
All right, video, if you want to come in a little.
Sure, I'll come in for a partial.
Get him back in. Oh.
Going a bit further.
Hey, Daniel, it looks like we're looking at an ophidioform or a cuskeel. I have not seen this one before, though. It's new to me.
Yeah, I'm also going through the gallery here, and I'm not seeing it. So, yeah, I know Bruce is looking intently at this.
That pelvic fin.
Yeah.
This is confusing. The pelvic fin isn't filamentous like the ophidioform. So this looks.
Yeah. Thanks.
This is a bathygatid. It's got a very tall second dorsal fin.
Oh,
It's probably in the genus Bathygatus, which we haven't seen before. My memory's failing now for the other more elongate genus that we've come up. I'll come up with something in a moment.
Copy that.
Thank you, Bruce. Yeah. Yeah.
Maybe a touch more.
Yes, this is unusual. You know, the more video close-up we can get, the better of it.
There's also a little
It
pollen.
would be good also to turn around if you could see if there's a barbel on its chin.
Watch lead. How much more of the ship's moving? So
Yeah.
the sponge, we might have to move on.
Yeah, let's take a quick look at that sponge. We just have a ship move. And yeah, as we were zooming out, you could see a nice pollinod scale worm right nestled on one of those rocks.
There's another fish.
So, Daniel, this bathygatid may be something similar to what we've got in the Hurl guide as Gadomus species only, and that's also CF. We're not really sure it's Gadomus, but maybe Bruce and Andrea got a good enough look where they could try to track this down.
Excellent. Yeah, thanks, Chris, for providing that. Yeah, it's definitely not in among the images of animals we got last year.
Where are we on the ship move, Nev?
So yeah, we'll try to get a good view here for you, Chris. We're in the middle of the ship move, but we'll...
Meter or two away. I mean, we're real close. Alright, copy.
Video, if you want to do a quick zoom.
Sure.
Yeah, thanks. This sponge that we've seen before always looks like it's a mix of live parts and dead parts, so I don't really understand what's going on with it.
Yeah. Yeah, definitely. You see different colorations and.
Can we zoom down just a little bit to see that part where there is some oscula?
Yeah, right there.
Perfect.
Sorry, video. Let me get it.
No problem.
And
Boy, I have no idea. I'm not even sure we're looking at a hexactinellid. Yep. Who knows?
Yeah, thanks, Chris. And you do see a little shrimp maybe on there. Some hydroids. Depth right now is 1,210 meters. The oxygen concentration is at 1.1, so where we landed closer to 1,300 meters, it was 1.5, so we're seeing it decrease. So yeah, there's this layer here, well, throughout the world, oxygen minimum layer between about 400 to 1,000 meters, where the oxygen concentration gets very low. Here in Hawaii, it's between about 700 and 1,000, and it's going to be very interesting that throughout this dive we're going to be traversing straight through it and seeing what effects that has on biological communities in many places throughout the world. There's a decrease or a general decrease in the abundance in life, but that's not true for all animals. Some animals actually do better in these environments, and that's been attributed to the fact that they might be able to tolerate these conditions better than their predators. To the right, we just passed a Bersingid sea star.
Ship is stopped, so you have can look around. We're in a safe spot. You're muted, by the way.
thanks, co-pilot. I'm going to set down and then we'll switch out.
And pilots, just so you're aware, it is about 800 meters to our second waypoint. So we have quite a distance.
Yeah, copy that. Nav, you're going to have to keep us moving.
Yeah,
so if we start lingering too long, start poking us.
Copy that.
Yeah, we absolutely want to make it to that top if possible. So we're just going through a quick personnel change here. And we also just completed a ship move, so we have a couple
Carl,
minutes here in this.
We can start moving the ship again.
Pilot, I was just telling them that we do have 800 meters to our waypoint too. So.
Copy that.
Yeah.
Just get comfortable here. Okay. So here we have a rock in the port box, and that's all?
That's correct, pilot. Yeah, so we did collect a rock sample at the very beginning of the dive. And so, yeah, I think we would like to cover some ground as much as possible. Yeah, definitely make that a priority to make it to the top of this ridge.
Sure. And what direction have our ship moves been now?
We've been going about, well, the last one was 45, but they averaged between 15 and 45, so our destination's about at 20-degree bearing.
Okay.
It looks about my heading right now is upslope.
Yeah, in the sonars.
Yeah,
so yeah, let's do another. We'll keep doing 30s at following the ridge. So now it would be 050.
Copy that. I'm going to call it into bridge.
I'm going to adjust your lower swing arms.
Sure.
And I'm going to just bring them out slightly.
Okay.
Bridge is nav. Hey, I'd like to request a ship move, another 30 meters at bearing 050 at 0.2 knots. Good copy.
So yeah, Nav, we'll do these 30-meter hops, and if all looks clear and we're not stopping for a zoom or a sample, then a few minutes after the ship stops, Sirius will stop swimming.
Hi, Daniel.
We can just call the next one.
We left it on.
So it'll be almost continuous, but we'll always have the option to stop and do what we want every 30
Yeah, we're
meters.
just going through a quick personnel change here.
Before I call it the next move.
But yeah, we hear you loud and clear.
I think waiting a few minutes is good.
So I was just updating our position here. So we have covered just shy of 150 meters in horizontal distance throughout the dive. So we landed on the bottom about an hour and a half ago. That was about 100 meters shallower than we are now, and about 150 meters away from where our current position. So yeah, this ridge runs to the top of the seamount. There's a kind of flat plateau area of the seamount that's at around 700 meters. And yeah, so we're just coordinating here to the ship moves and to try to cover some ground to try to make it to the top.
Copy that.
So do we have a ship move in now?
So yeah.
Yes. We're currently going
here along this ridge. So where we landed on the depth on the bottom, that was just off this ridge. So we were where we landed was just east of it, and there was no animals. There was very few of them. And then as soon as we started making our way over to the ridge, we see a nice aggregation of primnoid corals that seem to be consistently. And there's another fish to our left if we have a second.
Sure. It's a fast one.
There was actually looked like another fish on our right side too. Yeah. Same species maybe, or family, I guess.
So the fish to the left was pretty fast. I don't see it anymore.
Yeah, I don't either.
I'll scan over to the right and see what's over there.
Oh, and there's our first Metallogorgia.
Would you like a close-up of this?
Yeah, let's just get a quick snap zoom. I think it's the first time we see it.
Okay. Video. You can start going in. I'll just try to hover here.
Sure thing.
Come up to it. Hold there.
So yeah, that's Metallogorgia melanocristus, another one of these conspicuous components of the fauna at these depths. There's a few species, a few different groups that have this kind of
good. Hold there.
Plume and different morphology, where you have just all the polyps on this plume. There's black corals that have this, several Gorgonian corals.
Coming back up.
But again, that's one of these adaptations that getting off the substrate, because the polyps there on the plume are the ones that are feeding. And it's also an adaptation because it allows you to feed in kind of different flow regimes. So depending on where the currents come from, the plume can rotate around, and it's kind of like a windmill that will adjust its position depending on where the current comes from.
Can go wide.
And so it's an adaptation to feed efficiently.
They just wanted a snap zoom.
Sounds good. Thanks, Tyler.
Yeah, thank you. I know that was hard to focus with me bobbing around. Looked pretty good.
I do also want to take a second again to welcome all of our viewers that are watching this at one of our public viewing displays. Several have been set up around the state of Hawaii as well as in other places, including the Mokupapapa Discovery Center on the island of Hawaii. This wonderful facility we have in Hilo where you can learn more about the Papahanaumokuakea and its valuable resources. Also at the Waikiki Aquarium, as well as on the Maui Ocean Center, all of these premier educational facilities that we have throughout Hawaii. So welcome.
Say the same thing but disagree.
I just want to point out the nice landscape view that we've got right now. You can make out some faint layering again in the different layered cake type of form that we're looking at. Those are all different lava flows that have have been constructing this slope, and you can also tell that they're kind of dipping in the video from more or less the upper left-hand corner to the bottom right-hand corner.
Pretty linear in my.
That's basically pointing up to the rift zone ridge, which is the ridge that we're trying to get to right now. So we're traveling parallel to this ridge axis right now, but all of the lava flows that have erupted making this volcano up have been sourced from the ridge that we're heading perpendicular to. So very interesting morphology, and even though that this seamount has been around for tens of millions of years, letting the manganese crust kind of grow on top of it, cementing it together, it seems like the powers of erosion have revealed some really interesting primary structures. So
it's almost
very cool landscape that we're getting to observe today.
Do you still see it?
And then also interesting community here, one dominated by these primnoid corals.
It's about center screen, maybe.
And a nice moderate density here going along this.
Don't see it.
So yeah,
I see it now. Yeah.
There
it is.
Yeah.
Camouflaged.
So this is another one of these lizard fish, the halosaurs. And yeah, if we can take a quick look maybe on the head, and the scales on the head are important.
I can get in closer too if we want. If you hold their video, I'll just come in up and closer. Okay, you can go in video.
Aldo?
Yeah, it looks like an Aldrovandia. You don't really see scales on the head, which is a good characteristic to separate the genus Aldrovandia from another halosaur genus.
Yeah, excellent. Thank you. Thank you, Andrea. So yeah, these halosaurid fishes are very characteristic shape, and there's, as Andrea pointed out, two genera here in Hawaii that we have commonly imaged, and it's the scales on the head and how far they extend. That's main diagnostic feature to distinguish these two genera.
Thank you, Pilot.
Yeah.
Nice follow.
And as we're looking here on top, over to the side of this ridge, you see a nice aggregation going into a lot higher densities there on the towards the flanks there of that ridge.
Get out ahead before we do the next move.
Depth is 1,180 meters. And our oxygen, dissolved oxygen is around 1.1 milligrams per liter.
But the ship is stopped, right?
Just to put that into context, close to the surface, you would have levels around
coming up
7.2. And so we are close to this layer where you have low oxygen. Hawaii does not have a real oxygen minimum zone, which are defined to have oxygen contents less than 0.5 milligrams per liter. Our levels are slightly higher than that, so it's not a true oxygen minimum zone, but it's still a layer where you have very low oxygen concentrations, and it's going to be interesting to see how that shapes the communities at these depths. There we have another one of these stocked sponges, genus Sacocalix, very conspicuous for its shape. And a lot of these primnoid corals, which really dominate this faunal assemblage, mostly primnoid corals. We've seen scattered bubblegum corals and pink corals in between, but other than that, and so it looks like we have an Iridogorgia there. Yeah, that's a beautiful shot there. Maybe we can take a quick look if we have a second with the black background.
Yeah.
So that's our first Iridogorgia of the dive.
Get a zoom on it.
Yeah. Video, can we do a zoom here while I'm floating, when I'm moving closer?
Oh.
Yeah,
that's look like fishing net.
Yeah.
So yeah, that looks like some kind of fishing gear. So this The seamount is, of course, quite deep, 600